Monosaccharides can also form larger condensates, termed polysaccharides. Polysaccharides could consist of the same kind of monosaccharide residues. In this case, these are called homopolysaccharides, or being condensates of different kinds of monosaccharide residues, respectively called heteropolysaccharides. In turn, the residues, monosaccharide residues, could be linked either in strictly linear fashion or in branched fashion, respectively giving rise to branched and unbranched polysaccharides. As such, polysaccharides have two important biological roles as the energy storage, um, such as starch or glycogen, or as mechanical support, such as glucose or ketone. So there are also glycoconjugates between polysaccharides and peptides or proteins, forming, for example, peptidoglycan cell wall in bacteria. Let's look closer at some of these functions. An example of polysaccharides with energy storage function is starch. Starch is a mixture of two homopolymers of glucose. One is amylose. And amylose is a homopolymer of glucose where glucose residues are linked with one another by alpha 1 to 4 glycosidic bonds. Another kind of glucose polymer is branched homopolymer called amylopectin. Amylopectin consists of short amylose stretches where reducing end at some point is linked by 1 to 6 alpha glycosidic bond to another amylose stretch. This way, amylopectin represents a highly branched homopolymer of glucose with many non-reducing ends and very few reducing ends. The amylopectin can reach huge sizes containing up to several hundred millions of individual glucose residues. The amylopectin and amylose cluck together, forming microscopically visible starch granules. And the starch granules are well seen in chloroplasts of the plants, where chloroplasts fix carbon dioxide converted to glucose, and the glucose then is stored as starch granules. The animal analog of starch is glycogen. Glycogen is used to store glucose in animal tissues. In our body, the liver is very rich in glycogen. Similar to starch and amylopectin, the glycogen granules consist mainly of the branched polymer of the amylopectin, amylose-like stretches which are linked with one to one another by reducing ends, also by 1 to 6 glycosidic bonds, but the stretches are shorter, approximately 10 residues. In our body, tissues most rich in the glycogen are liver and skeletal muscle. In liver cells, the glycogen could constitute up to 7% of wet mass of the liver, and where it's used as a transit energy storage compartment. The advantage of storage of glucose in the form of glycogen is twofold. First is low osmolarity. If one would calculate the osmolarity of 7% glycogen stored in the liver, it would correspond to approximately 1/100th of micromole, which is minute. However, if one could convert all this glycogen into glucose monomers, it will be give approximately 4 mole of glucose, which will be huge osmolarity create huge osmotic pressure and would burst the cell. Another advantage is the maintaining glucose homeostasis. Glucose is can diffuse through the plasma membrane in our cells with the help of glucose transporters. By storing bulk glucose in the glycogen, liver cells could regulate glucose supply in the bloodstream, sequestering or releasing glucose into blood when necessary, keeping the glucose homeostatic concentration at around 5 millimole. An example of polysaccharides with the mechanical support function is another polymer of glucose cellulose. In cellulose, the glucose residues are linked not by 1 to 4 alpha glycosidic bonds, but by 1 to 4 beta glycosidic bonds. And this minute difference in stereochemistry results in a compound with completely different physical and chemical properties. In cellulose, the linear polymers of glucose is much larger, up to 7 to 15 glucose units. Within each of these stretches, the neighboring glucose residues are linked to one another, such allowing the most optimal configuration of hydrogen bonds binding them to one another. On top, this also allows optimal configuration of hydrogen bond between the different polymer stretches within the cellulose fibers. Because of that, the cellulose fibers have extremely high mechanical residues and are resilient to water, which underlies the function in plants as a principal component of the cell wall. The stereochemistry of beta-glucosidic bond in cellulose also underlies its remarkable resilience to remarkable chemical resilience, especially resilience to biological degradation. While starch and glycogen can be easily degraded to cellulose by alpha amylases in a number of organisms, including our body, there are very few organisms that can have equipped with enzymes to degrade beta 1 to 4 beta glycosidic bond linking glucose residues. Because of that, there are very few animals which can be fed with glucose as a food source. One of those are ruminants, and ruminants take advantage of symbiotic microorganisms if in the compartmentalized stomach in the rumen, which are in turn equipped with complement of enzymes able to degrade this 1 to 4 beta linkages between glucose molecule within cellulose. The animal analog of cellulose is chitin. Chitin is probably the second most abundant polymer on Earth. It forms exoskeleton of many invertebrates and the cell wall of fungi. Similar to cellulose, it is a linear homopolymer and consists of the 1 to 4 beta linked derivates of glucose called N-acetylglucosamine. The only difference between cellulose and chitin is the amino group in the second position of the glucose residues. An example of a heteropolysaccharide with mechanical support function is bacterial peptidoglycan. Peptidoglycan is a linear heteropolysaccharide consisting of two altering monosaccharide residues, N-acetylglucosamine and N-acetylmuramic acid, linked with one another by 1 to 4 beta glycosidic bond. On top, these linear chains are cross-linked by short peptide linkages. Together, peptidoglycan forms a mechanically rigid meshwork that protects bacteria from extracellular environment. As an interesting fact, the bactericidal properties of our saliva is determined by content of enzyme called lysozyme that can uh, kill bacteria by hydrolyzing these specific 1 to 4 beta-glucosidic linkages. Likewise, the bactericidal properties of antibiotic penicillin are determined by its ability to prevent the formation of peptidoglycan cross-linkages between the heteropolysaccharide and the peptides. An example of polysaccharides with mechanistical support function in our body are glycosaminoglycans, which are principal components of extracellular matrix in our tissues. All glycosaminoglycans are heteropolysaccharides consisting of repeating units of two monosaccharides, one of which is amino sugar and hence the name glycosaminoglycans. Different glycosaminoglycans have different composition. However, all of them are highly acidic, containing either carboxy sugar or sulfur sugar or both. Because of that, in conditions of normal pH, the residues in glycosaminoglycans are highly negatively charged, and this determines the property to assume highly extended conformation. This could be illustrated by the structure of the heparin segment, which is highly acidic sugar. It is assumes in solution a highly extended rod-like conformation. This property determines the ability of glycosaminoglycans to optimally roam the space between the cells. Different glycosaminoglycans, because of different sugar composition, have different properties and are predominant in different tissues. And often the trivial names reflect the tissues where they are predominantly found. For example, chondroitin sulfate, keratan sulfate, or heparin. To summarize, monosaccharides can form polymers which are called polysaccharides. Polysaccharides could consist of the same kind of units or different kinds of monosaccharide units, consequently being called homopolysaccharides or heteropolysaccharides. On top, they could form either linear polymers or branched polymers. All combination of these kinds are possible in biological systems. Polysaccharides mainly perform two roles: energy storage, such as starch or glycogen, or mechanical support function, such as chitin, cellulose, peptidoglycan, or glycosaminoglycans. The chemical and physical properties of polysaccharides and consequently their functions are determined by a combination of the stereochemistry of linkages between the monosaccharide units within polysaccharide and the chemical modifications and the chemical properties of the monosaccharide units.